I'm Sarah Feingold. Here's the buzz.
Imagine if you could transform from a couch potato to an Olympic athlete overnight. Well, honeybees actually undergo this transformation with a little help from the molecular level of their bodies. There are two stages in a worker bee's adult life, nurse and forager stage. After a worker bee is hatched, it serves as a nurse bee. Nurse bees work in the hive, performing tasks such as tending to the larva and queen and making honeycomb. Around day 23 in an adult worker bee's life, it undergoes a complete change in its muscle structure, and it becomes a forager bee. Forager bees work outside of the hive, collecting food. They can make up to 30 trips in one day and can carry 85% of their own weight. Obviously, they need incredible strength in order to carry such a heavy load, strength that nurse bees just don't have. In order to gain this massive strength in such a short period of time, bees change their DNA methylation, which is the way in which genes are expressed. Bees control how their genes are expressed through alternative splicing. So, what exactly is alternative splicing? Well, our genetic material is held in our DNA, which can be found in the nuclei of our cells. Think of DNA as the instructions to make a certain protein. In order to express a gene, or assemble the protein that DNA has the blueprints for, the DNA is transcribed into mRNA, which is translated into a sequence of amino acids which code for a specific protein. However, this process isn't as simple as it sounds. Not all of the DNA is transcribed into mRNA. There are some parts of the DNA that are cut out before transcription occurs. These are called introns, and the parts of the DNA that are left in are called exons. When transcribing a segment of DNA, the introns that need to be cut out have splice sites on each end. These splice sites are recognized by the machinery transcribing the DNA, and the intron will be cut out. However, sometimes introns are not spliced out. This changes the mRNA sequence that is made, which changes the protein that is produced. An intron will be retained in the sequence if a protein we call an alternative splicing factor obscures one of its splice sites while transcription is occurring. This process is known as alternative splicing. As we speak, there is research being conducted about alternative splicing around the world. If we can determine how it works, then we can figure out how honeybees control their genes, and maybe one day we'll be able to control human genes too. Thanks for watching!